This is how cells divide by meiosis to produce gametes, or eggs and sperm. We're going to start with a cell that's in interphase. Now remember, interphase is all of the cell's lifetime except when it's dividing, so that would include the G1, S, and G2 phases of the cell's lifetime. We're going to, instead of in human cells, which would have 46 chromosomes, just because it would be a little crowded and I don't have enough colors to draw that, I'll use a cell with 6 chromosomes. Here's the nucleus inside the cell, totally not drawn to scale. So here's a straight red chromosome and a squiggly red chromosome, straight blue chromosome and a squiggly blue chromosome, and a squiggly green chromosome and a straight green chromosome. Now imagine on each of these chromosomes, these chromosomes are made out of DNA. So there are genes, pieces of DNA along these chromosomes that are the instructions to build things. So say for example on the red chromosome, maybe at this locus or location, and also this one, since both red chromosomes are the same, we could have a gene for, say, hair color. But perhaps even though it's the hair color gene, the version of that gene on the straight red chromosome might be dark hair, but on the squiggly one, blonde hair. Those different versions of genes are called alleles. They still both make hair color, so it's the same genes, the same instructions, slight variation though. Say for example another gene might be at this locus, maybe that's the eye color gene, and it could have, say, brown eyes on the straight red chromosome, blue eyes on the curly red chromosome. The other chromosomes, they could have other information, you know, genes for finger shape and cleft chin and free earlobes and hitchhiker's thumb and tasting PTC, all those other traits. During the S phase, the chromosomes duplicate, so they're starting to copy the DNA. They're connected together at the centromeres, so there they are, all copied. We started out with a cell with 6 chromosomes, had 2 of each chromosome, 2 sets of chromosomes. So we're going to call that a diploid cell. Diploid means two sets of chromosomes. Sometimes that's abbreviated 2N. Okay, the diploid number would be 6. Now the point of meiosis is you can't use these diploid cells to reproduce with in sexual reproduction. For example, in people have 46 chromosomes in our cells. You can't have a woman with 46 chromosomes, marry a man with 46 chromosomes, and have a child with 92 chromosomes. That wouldn't be human. So there has to be a way to divide the cells so they get half the chromosomes in an egg and half the chromosomes in a sperm. So cells that have half of the chromosomes have only one set are called haploid cells. We're going to take these cells, this cell in interphase that would be in the gonads, either the ovaries or the testes, and go through meiosis. The first phase is going to be Prophase One of meiosis. The nuclear membrane starts to dissolve. Spindle fibers start to grow towards the chromosomes and eventually attach. Chromosomes condense, and let me show one difference between Prophase One of meiosis and Prophase of mitosis. If you look at this cell in Prophase One of meiosis, the big difference between that and Prophase of mitosis is that the similar chromosomes have paired up together. Similar chromosomes that have the same genes in the same locations, they're called homologous chromosomes. And these homologous chromosomes pair up during Prophase One. The next phase is Metaphase One, and in this phase, all of the homologous chromosome pairs move to the equator of the cell and are attached to the spindle fibers. The next phase is Anaphase One. In Anaphase One, the tetrads, or homologous chromosome pairs, split. The centromere splits at the middle and opposite chromosomes move to opposite sides of the cells. Now I'm not drawing the spindle fibers in just because the most important part is the information in the chromosomes. Notice how they're consistent and they're moving to the correct sides of the cell. The next phase is Telophase One. And in Telophase One, the nuclear membrane starts to reform and the chromosomes are divided. During Telophase One, the cells also start to divide in a process called cytokinesis. Or since this is only halfway through the process of meiosis, sometimes it's called interkinesis. And then the cells completely divide. Nuclear membranes are formed. Notice we still have 6 chromosomes, but they're not the same in each cell, and they're not the same as the chromosomes we started with in the mother cell to begin with. But we've not yet cut the chromosome number in half. We still have 6 chromosomes in each cell. So dividing those cells in half should get us down to 3 chromosomes each, or half the number. That's what we need in meiosis. So that's going to be the second half of meiosis, starting with Prophase 2. The nuclear membranes start to dissolve again. Spindle fibers start to form and attach to the chromosomes. Then the cells move on to Metaphase 2. In Metaphase 2, again the chromosomes line up along the equator of the cells, and the spindle fibers are attached, ready to pull those chromosomes apart, or the chromatids apart to opposite sides of the cells. Remember, half of a chromosome is called a chromatid. Now the next phase is Anaphase 2, and that's the phase where the centromeres divide and the chromatids are pulled to opposite sides of the cells. During Telophase 2, the nuclear membranes begin to reform, and the cell starts to go through cytokinesis again and divide. The chromosomes begin to unwind into chromatin, so they're not visible under a microscope anymore. And finally, when the cells finish dividing, that's the process of cytokinesis. The result is 4 different cells. Each of the 4 cells can form a gamete. Notice that the cells are different than the original cell to begin with. They're no longer diploid, they're haploid, only having 1 set of chromosomes each. Notice the top 2 cells are identical to each other, the bottom 2 cells are identical genetically to each other, but the top cells and the bottom cells are different. This is how meiosis results in genetic diversity. Now there's another process that even increases the amount of diversity, the number of possible kinds of chromosome combinations in these cells. Let's go back and look at Prophase One. Now during Prophase One, during Prophase One, chromosomes line up as a homologous chromosome pair, or a tetrad, and possibly as a mechanism of DNA repair, enzymes comb along these chromosomes looking for mismatches or damage or missing segments, and so they'll often break pieces of these chromosomes to compare the DNA. If pieces of these chromosomes were broken off and then reattached, but reattached backwards, you get the process called crossing over. So for example, imagine on the red chromosome, the 2 inside pieces, inside chromatid pieces broke off, switched places in a process called crossing over. This way you'd have half a straight red chromosome, half curly, then the other one half curly, half straight. And if we follow that through to the end, look at what difference it makes in the gametes. The difference in the gametes at the end is, for example, the top cells, the one with the straight red chromosome would have the instructions for dark hair and brown eyes. The one with the squiggly red chromosome, would have blonde hair and blue eye instructions. The one that's part straight, part squiggly would have dark hair but blue eyes. The one that's part squiggly and part straight would have blonde hair and brown eyes. So we've doubled the amount of variation just by doing one crossover event. And when you think that several chromosomes during meiosis, during Prophase One, can go through crossing over, or even cross over in multiple places along the chromosome, the amount of genetic variability is amazing. It's astounding. Virtually no one, I mean in all reality, no one on this planet has probably ever had the exact genetic combination that you do. Every individual is genetically unique. And this process of crossing over also explains why some traits that normally seem to go together sometimes don't. For example, blonde hair and blue eyes seem to go together. Most people with blonde hair seem to have light colored eyes. However, once in a while you have someone with blonde hair and brown eyes, or dark hair and blue eyes. And so these kind of traits that normally seem to go together often are called linked genes. They're on the same chromosome and tend to go together as a package, but through the process of crossing over can be reassorted. And that's how meiosis occurs to produce genetic variability for gametes.